Hey guys, this is Professor Tiffany Martin with Medical Microbiology, and we are in Chapter 10, which is controlling microbial growth in the body. So basically chemotherapy drugs. We are working our way through mechanisms of action. How do these drugs work? So we so far have looked at we're looking at 5 different categories. We looked at the first category, which is cell walls. This is where the mechanism of action of a drug will inhibit the synthesis of cell walls. Usually because it doesn't let the cell walls connect with those peptide cross-links from that. Because humans don't have cell walls, the selective toxicity is good. These drugs don't hurt us, so they typically are safe, very few side effects, they're very good. Now we're going to move to this category of protein synthesis. Now this one's a little tricky because we synthesize proteins too. So bacteria synthesize proteins, but so does the host. So we need to be careful about that. But there are some there's some good news on this, and there is some ways that we can target prokaryotic protein synthesis without harming eukaryotic or the host protein synthesis. Okay, so talking about protein synthesis, we're talking about translation, right? You remember that? mRNA goes to the ribosome will translate the mRNA into a protein synthesis. So we're talking about the concept of translation. So if you struggled in that DNA section with translation, you may need to go back and review just a little bit on the basics of how translation works. All right, selective toxicity is actually relatively good. Because prokaryotic cells have ribosomes, eukaryotic cells have ribosomes, that's true, but there are enough differences between our prokaryotic and eukaryotic ribosomes that we can have drugs that harm prokaryotic ribosomes without harming eukaryotic ribosomes. So what do they mean when they're different? Let me tell you just a little bit about these ribosomes. The ribosomes, if you remember right, you've got the large subunit and the small subunit, and that comes together on the mRNA to get translation. And that's true for both prokaryotes and eukaryotes. However, the ribosome, those subunits are made of proteins and some RNA, ribosomal RNA in there. There are enough differences that these ribosomes, even though they do the same job and have the same function, they're slightly different. And we know they're different because when we isolate them from prokaryotic cells and we isolate them from eukaryotic cells, they have some different qualities to them. So we can see they're different at the molecular level, and then at the proteins, when we can actually centrifuge them down. There's a way that you can have you can centrifuge them spin them at a rate, and you can actually measure how far down they sediment based on the speed of centrifugation. And so these are the units of measurement that they use are called stuvarts. I don't care if you know this very much. I don't even think I'm pronouncing that right, but it's an S measurement. And so the ribosomes that we isolate from prokaryotic cells are what we consider 70S ribosomes. They sediment out at that 70 mark, that 70 Stutvert mark from that. The ribosomes that we isolate from eukaryotic cells, we refer to those as 80S, and it's just because they're slightly different. They're slightly larger, and they sediment slightly differently. All right, so the book has a little bit more explanation of that. You can look that up if you're interested. But basically, what you need to know is that the ribosomes from prokaryotes are slightly different, and different enough that we can target prokaryotic ribosomes and leave our eukaryotic ribosomes alone. So in regards to selective toxicity, that makes this category good. I can't do this. Makes this category good. Now, what I wanted to tell you to be careful about is our eukaryotic cells. We have ribosomes. But if you remember back to your cell structures of a eukaryotic cell, we have mitochondria in our eukaryotic cells. We have chloroplasts in our plant eukaryotic cells. The mitochondria have its own DNA, if you remember that. They also have their own ribosomes in the mitochondria. So again, you can go back to that endosymbiotic theory. We think that they were cells that kind of merged, but either way, mitochondria have their own ribosomes. And the ribosomes in our eukaryotic cells in the mitochondria have a lot more similarities with our 70S prokaryotic ribosomes. So we can target those prokaryotic 70S ribosomes, but we need to be careful that they don't interfere with our host mitochondrial ribosomes. So that kind of puts it in the good category, but be careful because of these mitochondrial ribosomes. Okay, so selective toxicity generally good because we can target the 70S one. What we need to be careful about is the mitochondrial ribosomes look very similar to bacterial ribosomes, so we have to be careful about that. All right, so let's look at some drugs in this category. We're going to look at 4 different drugs. These are in purple, so kind of remember these, and how they interfere with protein synthesis, translation. So we're going to go back and forth to these 2 slides. Streptomycin causes a shape change of the ribosome, which results in reading the mRNA incorrectly. Let's go back to some basic translation. Here is my large subunit. There is my small subunit. Here is my mRNA that goes through. You remember how our tRNAs come into these windows. They come into that A window, get moved to the P window, and then they leave through the exit window. You get this growing polypeptide chain. Again, if you need to go back and review that to make sure you understand translation, you can do that. All right, so how do these drugs interfere with this process of translation and this making protein synthesis, making of this growing polypeptide chain? All right, so here's streptomycin and other drugs like that. They will bind here. And you can see when they bind, instead of the mRNA going through smoothly, it actually creates kind of a kink in that. That interrupts my tRNAs. When they come in with their anticodon to match up to the correct codon on my mRNA so they can bring in the correct amino acid, because it's bent, different tRNAs can come in here and bind that shouldn't bind. That means I'm making a protein, and that protein doesn't necessarily have the right amino acid sequence. So these amino acids are going to be different, then they're going to fold different, and that protein is not going to work. So how does streptomycin work? It binds in this to the mRNA, causes a change in shape, and that allows tRNAs to come in and fit that don't match the codon. And I'm going to make a non-functional protein. I'm going to make a protein. I will make a protein, but it won't be the protein that I wanted to make. All right, so that's how streptomycin works. But there are other drugs that work in different ways. Tetracycline, this is a drug you might have heard of before. This one actually blocks the attachment of tRNA to that A site. So if we look at the picture on this one, here is my mRNA like it's supposed to, my large subunit, my small subunit of my ribosome. Everything looks good. I've got my growing polypeptide chain. There's my P site with my growing polypeptide chain. Here's my A site, but my A site is blocked. Tetracycline came in and bound in this A site, and now it's blocking any more tRNAs from coming in. In this case, I make a truncated protein. Whenever this tetracycline binds, and it's going to be competitive inhibition, if it's just as soon as it just depends on how the dosage of the drug, how much tetracycline is there. Proteins will get started maybe, but they won't be able to finish. So that is how tetracycline interferes with protein synthesis. And other drugs, this is chloramphenicol. This one actually prevents peptide bond formation. So going back to the drawing and looking at that one, here's my mRNA and my ribosome, and everything is working great. My ribosome is going through, the correct tRNA is binding to my codon, and it's bringing its amino acid, but instead of being able to join these amino acids together, chloramphenicol prevents this peptide bond formation. So protein synthesis thinks it's going through, but instead of making this long connected polypeptide chain, those amino acids just get released as individuals again. I always think of this one like a toddler on an Easter basket. Have you guys ever watched a toddler doing an Easter egg hunt? And they've got their Easter basket, and they pick up and they find an egg and they put it in their basket, and then they lean over to get the next egg, and all their eggs go out of their basket, and they pick it up and they're like oblivious. They're just keep doing that, and their eggs keep doing that. That's my visual in my head for that one. So do I make a protein for this one? Nope, no protein gets made on this one. Synthesis proceeds, but we don't end up with a polypeptide chain with chloramphenicol. Okay, I got another drug, erythromycin. Erythromycin is going to stop the ribosome from moving along the mRNA. So what's going to happen in this one? Here's my ribosome, here's my mRNA, and then I have a drug here that's going to bind and stop this ribosome from moving. So unlike chloramphenicol, where the ribosome keeps going and we just don't get a peptide made, this one actually blocks this and it doesn't move. We might get a truncated protein, a shorter protein from that, but that's not going to work as well. All right, these are some ones at the bottom. I don't have any drugs. I didn't make you learn any drugs for this one. This one is kind of interesting because now that we know, we can easily sequence these proteins and know exactly what their DNA sequence is. We can actually engineer a sequence, a nucleic acid sequence, that's complementary to mRNAs. These can actually serve as drugs. We put in this sequence of nucleic acid. It doesn't have to be very long, it just has to be long enough to stop this process. It's going to bind to the mRNA, and then I don't get translation. This is a drug that they're working on for Ebola. I don't know how far it's gotten, but this is one of these drugs. We can actually design drugs. So your question is, well, let's just design drugs for everything like this. This takes a lot of work to come up with this sequence. It's also very specific, which can be good or bad. It's only going to work against one pathogen because it's only going to have that complementary base pair against that one pathogen. And this one is easily, mutations can occur and then that drug won't work anymore. So I spent all this time and money designing this drug, and then the pathogen has some mutations and the drug doesn't bind anymore, and now my pathogen is resistant to this drug. Okay, but there are some out there and they do work for that. Okay, so here's my examples. All of these are in the category of blocking protein synthesis, and all of these are in regards to selective toxicity. It's it's good because we can target prokaryotic protein synthesis with those 70S ribosomes, but we need to be careful because we don't want to interfere with mitochondrial ribosomes. Okay, so I'll do cell membranes in the next video.